Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Both human and yeast cells use glucose as an energy source, but variations in biochemical pathways produce different products. Point 1 Variation in a biochemical pathway that occurs in a human cell is shown below. The biochemical pathways for fermentation in human and yeast cells share the universal starting point of glycolysis, which converts glucose to pyruvate and yields ATP under conditions lacking oxygen. The crucial difference lies in the final products and associated enzymes. Human cells produce lactate through a single enzymatic reaction, while yeast cells employ a separate set of enzymes to yield ethanol and carbon dioxide. As this is a compare question, similarities and differences, or a combination of both, should be provided. Enzyme assistants. They help enzymes lower the activation energy of a reaction, but do not catalyze the reaction themselves. Carrier function. They act as essential carriers, specifically transporting energy, ATP, protons and/or electrons, NADH, FADH2, NADPH. Cyclic activity. They continuously cycle between their loaded form carrying the atom energy and their unloaded form, ensuring the pathway can proceed efficiently. Don't confuse the role of coenzymes with that of enzymes. Coenzymes do not catalyze reactions, instead, they assist enzymes in their role as protein catalysts. The World Vegetable Center, World Veg, collects, conserves, and identifies important traits in vegetable plants. With this information, scientists are leading an initiative to increase both crop diversity and crop yield in Africa. The scientists involved in World Veg investigated many different vegetable crops. They employed advanced technology using an automated laser scanner. This scanner moves over fields of vegetable crops to capture 3D models of the plants under various scans plants 3 times a day, collecting as much data as 400 field workers would collect in a 24-hour period. The scanner can emit different wavelengths and intensities of light. The wavelengths reflected by the plants are one way in which data is obtained for analysis. The scanning technology enables the monitoring of many plants and helps identify those with desirable traits for vegetable crops. Plants can be exposed to different conditions, such as flooding or heat stress, and the scanner can assess individual plants' responses in real time. Plants responding well to the changing conditions can be identified, leading to the development of improved vegetable crops that are either heat or flood tolerant. Heat tolerance in a plant is a characteristic that results from a combination of traits. Scientists need to study many traits simultaneously to identify those traits that make a plant heat tolerant. Using the scanner, scientists evaluate 75 different traits, including plant height, biomass, and leaf characteristics, throughout the plant's life cycle to select heat-tolerant varieties. Without the scanner, scientists would have to rely on only a few traits for selection and may miss important plant features. Once plants with desirable traits are identified, the plants are grown, and seeds are collected. In Africa, the collected seeds are being distributed to African farmers. The farmers are encouraged to grow the seeds and produce crops with a higher yield. Scientists analyze reflected light wavelengths to gauge a plant's biochemical activity. Key information is derived from understanding that poorly absorbed wavelengths are highly reflected. This provides data on photosynthetic efficiency, identifying the wavelengths most critical for light absorption by pigments, like chlorophyll, during the light-dependent stage. Biomass and health, quantifying the amount of light absorbed, which correlates directly with the concentration and type of chlorophyll and the overall health of the plant growth stage, determining if the plant is in a specific production or flowering phase. Two key pieces of data, other than reflected wavelength, used to determine vegetable crop suitability include variable crop yield, such as final biomass or overall output, and the plant's observable growth characteristics, including height, leaf quality, and the overall resistance to environmental stresses like heat or flooding. The scanner offers key advantages over traditional fieldwork, primarily by allowing for faster and more comprehensive data gathering, collecting a greater variety of plant traits. This speed and depth of information, in turn, facilitates the easier and more efficient identification of desired plant varieties, which directly supports farmers in their efforts to maximize crop production and improve their livelihood. Photosynthetic pathways in C3 and CAM plants share the use of rubisco for fixing CO2 in the Calvin cycle and perform the light-dependent stage before the light-independent stage. They differ fundamentally in timing and initial enzyme. C3 plants fix CO2 directly with rubisco during the day, while CAM plants use PEP carboxylase to fix and store CO2 as malate at night. This temporal separation is a crucial adaptation for hot, arid environments, as it enables CAM plants to close their stomata during the day, thereby minimizing water loss and reducing photorespiration at high temperatures, which in turn ensures their survival. For one of their VCE biology practical activities, a group of students investigated the relationship between biomass and stored carbon in different tree species. The biomass of a tree is a key indicator of how much carbon in the form of carbon dioxide, CO2, a tree has captured from the atmosphere. To estimate how much CO2 a tree has captured, the students conducted fieldwork to measure the biomass of different trees. The students used a measuring tape to record the tree trunk circumference, which they then used to calculate the diameter of each tree. The students' method is outlined below. Method: identify the tree species. Measure the circumference, cm, of the trunk at a height of 1.4 meters. Calculate the diameter of the trunk in cm, refer to the provided graph to estimate the tree's biomass, kilogram, based on the calculated diameter, cm. The effectiveness of black bud at capturing carbon dioxide is justified by its capacity for high carbon fixation and storage. This capacity is evidenced by the species demonstrating the highest predicted biomass compared to other species, meaning it converts and stores more atmospheric CO2 through photosynthesis. Furthermore, data analysis confirms a positive correlation between trunk diameter and biomass, with specific measurements, e.g., 1,900 kilograms biomass at 50 centimeters diameter, supporting its superior growth rate and ability to sequester carbon. The methodology suffers from limitations concerning both reproducibility and generalizability. Measurement flaws: the calculation of biomass is flawed as it relies only on trunk diameter and assumes an ideal circular trunk shape, ignoring other significant biometric components, e.g., overall tree height, root mass, or the number of leaves or branches, which greatly contribute to total carbon storing biomass. Scientific rigor: the study lacks scientific rigor, as there is no indication of repeatability, how many trees should be measured, nor a clear protocol for species identification, which limits the ability of other researchers to replicate the study or generalize the comparison beyond the limited number of species examined. Safety precautions: awareness of surroundings. Students must be vigilant for tripping hazards or uneven ground and should avoid standing directly under large branches that could fall. Protective gear: Appropriate protective clothing must be worn, such as sturdy boots, gloves, and a hard hat, depending on the site, to minimize personal injury risk. Ethical guidelines. Environmental respect. Students must ensure that living organisms, or the ecosystem, are not disturbed or damaged. This includes refraining from removing any material, such as leaves, soil, or bark, during measurements. Compliance and permission. Students must obtain approval from the relevant authorities, such as the park or forest management, before conducting any fieldwork to ensure the activity is sanctioned and appropriate. CRISPR-Cas9 technology can improve photosynthetic efficiency by engineering two critical components: chlorophyll genes, altering genes to boost chlorophyll production and increase the rate of light absorption. Enzyme genes, such as Rubisco, modifying the gene for Rubisco to enhance its CO2 specificity, thereby reducing wasteful photorespiration and increasing the overall efficiency of the light-independent stage. Cellular respiration and fermentation. ATP control. Glycolysis rate is inhibited by high ATP levels, etc. Limiter. Oxygen is the immediate limiting factor for the electron transport chain. Fermentation. Both yeast and human cells use glycolysis, but the final products differ. Photosynthesis and adaptations. Process link. Light-dependent reactions in the grana produce ATP and NADPH, which power CO2 fixation in the stroma. Water role. Water acts as the electron donor in the light-dependent reactions. CAM adaptation. CAM plants minimize water loss by separating CO2 fixation in time, night versus day. Biotechnology and fieldwork. Biotech goal. CRISPR targets chlorophyll for light absorption, or Rubisco, to reduce photorespiration, to boost efficiency. Scanner use. Automated scanners collect massive data to identify stress-tolerant crops. Methodology flaw. Relying on trunk diameter is limited, ignoring major components, e.g., height or root mass, and assuming a circular trunk. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.